Thank you very much, everyone, for joining us for our fourth webinar in this series. It's on synapse-to-nucleus signaling. I need to express the regrets of my colleagues Michael Greenberg and Christine Holt, who couldn't be with us this afternoon, but I'm very happy to welcome as co-moderators Paul Donlan-Asp, who's an assistant professor at the University of Edinburgh, and Chao Sun, who's an assistant professor at Aarhus University. You'll hear from them when they introduce our speakers, who today we're really thrilled to have in this session: Mike Fainzilber from the Weizmann Institute, Yingxi Lin from UT Southwestern, Dallas, and Dick Chen from NYU. The format of the webinar is the speakers will go in the order of Mike, Yingxi, and Dick. Each of them will speak for around 25 minutes. And after each of their talks, we'll have the opportunity to answer questions. And if you have questions, as soon as they begin, you can start typing your questions into the chat. And then the moderator of that talk will read your question out and the speaker will answer it. Following the last talk, we'll then have a general discussion where we can revisit questions that we didn't have time for of individual speakers, and you'll have the opportunity to ask more questions, and we can even have a general discussion then. I believe that's all of the introductory stuff I need to say, so I think I'll hand it over to Paul to introduce Mike.
Perfect. So thank you. So as Erin said, so I'm Paul Donlan-Asp from the University of Edinburgh. So I want to start off by thanking both Erin and Martin for the opportunity to fill in as a moderator today. And it's actually a really great pleasure and honor that I get to introduce our first speaker for today's Brain Prize webinar. And it's because it's someone whose actual work has informed and driven my own research since I started in the field of RNA localization and developing axons.
There compared to the...
So Mike is joining us today from the Weizmann Institute of Science, where he heads the Department of Biomedical Science. So part of the fun of introducing such distinguished speakers is getting to do kind of a deep dive into what they've done over their career. And so actually Mike received his PhD from Hebrew University, where he studied and characterized mollusk-specific toxins from cone snails. And he would continue this kind of work on as a postdoc both in Amsterdam and then in Stockholm. But it would be really when he returned to Israel and started
by comparison
that he really dived into the intricacies of molecular and cellular mechanisms underlying neuronal growth, function, and most notably axonal regeneration. So his group has actually made really foundational contributions to our understanding of molecular responses underlying axonal injury. And his group has discovered that after axonal injury, important beta-1 is locally translated in injured axons. And we actually understand that this locally synthesized population of proteins is required in retrograde signaling from the damaged axon back to the nucleus, which is essential in axonal regeneration. His group has also gone on to demonstrate this axonal retrograde signaling of newly synthesized proteins is important just in general function of the neuron as well, finding it important in regulating mechanisms controlling cell size, axonal length, and basic neuronal functioning. And so kind of with that, I'd like to turn it over to Mike and look.
Thanks a lot, Paul, and thanks to Martin and Lundberg.
Yeah. Okay.
And thanks and congratulations to Erin and Christine and Mike for the well-deserved prize and for including us in this event. And without further ado, let me try and find my presentation and share it with you. Okay, can you see this?
Yes, we can.
Yeah, okay, that's good because you've all vanished and I can only see the presentation. So anyhow, so the session is about synapse-to-nucleus signaling, and I'm actually going to be talking about axon nucleus signaling and bidirectional axon nucleus signaling. So axon to nucleus, nucleus to axon. And we got into this topic, as Paul kindly mentioned in the introduction, first studying injury signaling from the axon to the cell body. And more recently, in recent years, the mirror image of that question, studying how axonal growth is controlled and how neurons can sense the length of their own axons. and I will very briefly review the history, some of which some students tell me is ancient history, which I object to because some of it was work I did. And this is a problem at my age to think I'm already ancient. But anyhow, I will briefly review the history, and then I will go into a new story, which is mainly unpublished data that I would actually really like feedback on in the chat, in the questions, or later on via email, anybody who's interested. So early on, when looking at retrograde injury signaling, as Paul mentioned, we discovered that important beta-1, the main cofactor in the important beta, important alpha nuclear import complex, is locally translated in peripheral sensory axons upon injury. And this triggers formation of a retrograde signaling complex linked to the molecular motor dynein that enables nuclear localization signal containing cargoes, such as transcription factors and other proteins, to move from the injury site in the axon to the cell body, to the soma, and also to the nucleus. And this gives you a direct route for communication from the axon to the nucleus. And one of the major insights from this early work was that both signaling, regulatory, and scaffolding components of the complex are all generated by local translation. And I'd briefly like to point out that much of this early work and also all of our ongoing work is in close collaboration with Jeff Twiss from the University of South Carolina. And Jeff has made really major contributions to understanding axonal local protein synthesis in peripheral sensory axons and also in other types of neurons. And he was instrumental in helping us actually understand early on that the appearance of important beta-1 in the axon was due to local protein synthesis, thereby introducing me to this field and basically helping us really understand this mechanism. So thanks, Jeff, if you're out there in the audience, and I hope he will get a chance to speak in one of these series very soon because he well deserves it. And just to briefly mention that while we were characterizing the roles of importance in injury signaling in sensory neurons, Kelsey Martin, who spoke in one of the earlier webinars in this series at UCLA, was describing the involvement of importance in synapse-to-nucleus signaling in aplesia and mammalian systems. Michael Kreutz at the Leibniz Institute in Magdeburg and his colleagues have also shown involvement of importance and related molecules in NMDA receptor signaling from synapse to nucleus. And colleagues at the Weizmann, Zivrach's lab and Michael Elbaum's lab actually showed involvement of importance in cytoplasmic transport in non-neuronal cells. So the mechanism that we're dealing with is not a very specialized injury-specific mechanism, but it's a general cell biological mechanism that integrates transport from cell extremities to cell centers, also in cycling cells and most definitely in neurons. And in more recent years, as I mentioned, we've been interested in the flip side of the injury signaling question, rather than asking ourselves how a signal from the end of the axon reaches the cell body. We're asking ourselves how the cell body actually can sense the length of its axon and regulate the growth of the axon. And I'm not going to go over all the work that went into elucidating this mechanism because it turns out to be the same mechanism. And to summarize a body of work over the past 10, 15 years, what we've shown in a series of studies is that growth and length sensing in peripheral sensory neurons and also in other neuron subtypes is dependent on a mechanism which functions by anterograde transport of RNA on a kinesin carried complex through the RNA binding protein nucleolin. RNAs associated with nucleolin are locally translated at the axon tip or at stations along the axon upon appropriate stimuli. And these RNAs include important beta-1 that I mentioned in my introduction, other well-known molecules such as mTOR that form a retrograde complex that moves from the tip of the axon back to the cell body. And this anterograde, retrograde RNA out, protein in system basically provides a scanning system for a cell that can be used for many different physiological outcomes, but primarily in the work we're looking at how this system is used to monitor or regulate growth of the axon. Now, when modeling this system, one of the elements that comes out of molecular modeling and trying to understand computational modeling of the system, one of the primary postulates comes out of this is there should be a negative feedback loop by which the retrograde signal represses the anterograde RNA transport. This would allow resetting of the mechanism, and this would actually allow the mechanism to sense length. And if anybody's interested in how, I can go into that later. But
Greenberg and Christine Holt, who couldn't
wanted to tell you today is the question, if this negative feedback loop exists, what might it be? What might provide negative feedback to this type of mechanism? And here is where I shift from the published history to the unpublished work that we're hoping you'll find interesting. So Christine Albus, when she was a postdoc in the lab, raised a very interesting hypothesis, and her idea was that alternative polyadenylation might be involved in regulating axonal localization of these RNAs. So alternative polyadenylation, as you know, utilizes the proximal or distal polyadenylation sites to put the poly A tail on an RNA. And since many axonal localization motifs are in the 3' UTRs of RNAs, if you're using a proximal polyadenylation site, that will give you an RNA that most likely is not localizing to axons, whereas if you're using a distal polyadenylation site, you will get an RNA that contains axonal localization motifs and should be taken out to axons. And Christine thought that perhaps there would be a switch between these types of polyadenylations, and that will give you switches between axon localizing versus non-axon localizing populations, and this would be a nice way to regulate the system. So this is a very cool idea. We thought it was a very cool idea. We invested a lot of effort in trying to obtain evidence for this. Christine life intervened. Christine moved to the UK. Indrek Koppel came from Estonia to do a postdoc. He's now running his own group back in Estonia. But he set up a workflow together with Riki Kawaguchi, a collaborator at UCLA, to do 3' end sequencing of injury regulated transcriptomes in sensory neurons to see if one could identify switching of polyadenylation site usage in this system before or after or during injury. And a lot of effort went into showing that Christine's hypothesis is wrong, that at least in our system, we see few, if any, polyadenylation switching events. But here is where wrong hypotheses are actually very valuable, because what this hypothesis required Indrek to do was to really look very, very carefully at upregulation of polyadenylation sites in the data sets he had. And one of the observations he made, which we would have not made by any other way, is that all of the apparent
following the last talk
he was seeing in the axon were due to upregulations of polyadenylated RNAs called signs, which I admit I had never heard of before Indrek came into my office saying, I figured out what we're seeing in the data. So what are signs, or in this case B2 signs? Signs stands for short interspersed nuclear elements. These are transposons in rodent genomes. You're probably more familiar with their closest analog in primates, the ALU elements. There are about 150,000 copies of B2 signs in the mouse genome. These are short non-coding RNAs. They have internal priming sites for RNA pol 3, so they're transcribed by RNA pol 3, and they're polyadenylated. And this is the secondary structure of a B2 sign RNA, and you can see that an ALU RNA basically looks like two adjoined B2 signs. So we were looking at this bizarre result and saying, okay, we need to figure out if this is specific, if this is real. Indrek looked at the data set. Indeed, when looking specifically at B2 sign RNAs in the data set, one sees a global induction upon peripheral nerve injury by different methods. He was able to confirm this by qPCR on specific loci. He was able to confirm this by Norton blot on sign RNAs using a consensus probe generated from a consensus B2 sign sequence. And I'd like you to remember this sequence logo because it will feature later on in the talk. And you can see that although there are some conserved sites within the signs, it's 150,000 loci, so there's a lot of degeneracy throughout the sequence. We were then fortunate to have access to data sets from collaborating labs, which are part of the Edelson Medical Research Foundation Consortium, which we're fortunate to be part of. So we asked Mark Tuschinski from UCSD to look in his data sets of DRG, sensory neuron injury, after sciatic nerve injury. And as you can see, mice injured in San Diego respond in the same way as mice injured in Rehovot by significant upregulation of B2 signs. We asked ourselves, is this happening also in other neuronal types? So likewise, through the Edelson Consortium, we have a collaboration with Zhigang He's lab at Harvard Medical School. Zhigang works in the optic nerve retinal ganglion system, amongst others. So we were able to look at a very comprehensive database that he had generated for RGC injury. And I'm clicking through as we speak, and I think you can appreciate that there is little or no regulation of B2 signs upon nerve crush in retinal ganglion cells, in stark contrast to the sensory neuron data I've just shown you. So this basically summarizes that data. You see here the upregulation of B2 signs in sensory neurons and peripheral nervous system, lack of regulation of these B2 signs in retinal ganglion neurons, central nervous system, and lack of any meaningful regulation of all the other repeat elements one sees in both of these data sets, and one sees many, many other elements. So this is a specific response of a specific class of non-coding repeat element RNAs in peripheral neurons after injury. Does this have any biological significance? And we thought to test this first by trying to overexpress B2 signs in the central neurons that do not upregulate them natively upon injury. And to this end, we generated a adeno-associated virus construct expressing a B2 sign consensus sequence. And Zhigang's lab injected this into their optic nerve crush system 2 weeks before the crush, ran an optic nerve regeneration assay 2 weeks later. And as I mentioned, we have a long-standing collaboration with Jeff Twiss, who's fortunately for us also part of this Edelson Consortium, and people in Jeff's lab, Elena Della Costa and Pabi Sahu
from cone snails, and you would continue this.
Rutgers were able to do the fish assays to show that this B2 sign construct is actually overexpressed in retinal ganglion cells. And this is not as trivial as it sounds, because you're trying to pick up a specific signal from a background of 150,000 ubiquitously low-level expressed non-coding transcripts that are basically in every possible cell in the mouse genome. So does this have a functional effect? Yes, it does. This is the regeneration one sees in B2 sign overexpressing retinal ganglion axons. And you can see the quantification here. I will say that this is not the kind of massive regeneration that Zhigang He's lab has shown for other interventions in the retinal ganglion system, but it is there, it is robust and replicable. We went back to Mark Tuschinski to try and do the similar type of experiment in systems that he works in. So corticospinal neurons injured by lesion of the spinal cord, and looking at regeneration or sprouting of corticospinal tract axons. Erna van Niekerk in his lab did these experiments. So again, the same AAV B2 sign construct injection into the motor cortex, spinal cord crush, and then regeneration assays. And as I hope you can appreciate, systems that receive the B2 sign show much more extensive sprouting than those that receive control constructs, and this is quantified here. So just to summarize this in cartoon form, we seem to have identified a non-coding RNA, a repeat element non-coding RNA family, which is upregulated specifically upon injury of peripheral neurons, but not central neurons. It seems to enhance regeneration, and upon overexpression in central neurons will also enhance regeneration of those neurons. So then the question becomes, how is this specific transcriptional induction occurring, and how is the enhancement of growth being mediated by these RNAs? So by this point, it's a 15-minute talk, but it's like 8 years of research gone into this project. By this point, Indrek's postdoc period was up. He was moving back to Estonia to start his own research group in Tallinn. And Eitan Zahavi picked up this project and is bringing it to the finishing line, we hope. And what Eitan decided, to answer the first question, was to get more detailed mapping of precisely which B2 signs were being upregulated in our system. So again, working with Ricky in UCLA and with Shifra Bendor and Esti Feldmesser in the bioinformatics core facility on our campus, we did resequencing to get longer reads of the signs in the system, mapped them to the genome, the mouse genome, which is a very non-trivial task, and identified precisely which loci were responding. And then we got a very striking result, because it turns out that of the 150,000 B2 sign loci in the mouse genome, only about 450 are being upregulated in injured DRG neurons, and the vast majority of these are in intergenic or intronic regions of the genome. So they are not being regulated due to adjacent activation of a coding gene. They are not embedded in the functional part of a coding gene. They're acting as independent transcriptional units in regions of the genome that classically would have been thought of as gene deserts. So it's a specific upregulation of a subset of the sign family that we're currently designating the GI signs for growth-inducing signs. We took advantage of an ATAC-seq dataset generated by Igor Ulitsky's lab at the Weizmann to look at these loci, and the GI sign loci are localized at injury-associated accessible chromatin in DRG. And when we were able to compare this to an RGC dataset generated in Jigang He's lab and analyzed by Yuyan Cheng at UCLA, we could see that these loci were again enclosed chromatin in the RGCs. So this explains why you see upregulation of these GI signs in sensory neurons but not in retinal ganglion cells upon injury. What controls opening or closing of these chromatin regions upon injury is something that would be interesting for the future, and we currently do not know. Looking more deeply into the GI sign loci and Igor axon to the cell body, we made another striking observation that actually these loci are specifically associated with AP1 transcription factor binding sites. So the AP1 transcription factors, June FOS, ATF3, these are very well known immediate early genes and also very well known as post-injury transcription factors in DRG. And we were able to confirm using a dominant negative FOS construct that expression of this dominant negative attenuates GI sign expression in DRG cultures. So you can repress this upregulation by repressing AP1 transcription factor activities. So this is an effect of POL2 transcription factors on a POL3 transcribed gene family, and it's another interesting aspect of the regulation here that would be worthy of further study. So we have a very specific gene family or subfamily of these non-coding RNAs that is being controlled transcriptionally by FOS-June transcription factors, but how is it actually doing its job in enhancing axon growth? And to get a handle on that, we took the GI sign sequences and synthesized them in basically in two halves of the sequence, the first 75 nucleotides and the rest biotinylated, and used these probes to pull down whatever might bind to them in axoplasm, in this case from sciatic nerve, and had this material analyzed for us by Juan Oces in Al Burlingame's mass spectrometry facility at UCSF. We've been collaborating with Al likewise for decades by now on many aspects of our work. And very strikingly for us, the proteins that came out of this, the top 20 enriched proteins, one of them is nucleolin, which I will remind you is the main RNA binding protein that takes components of the growth regulating RNA system out to axons. And the other interesting set of binders were ribosomal components. So, and actually, if you spread out this analysis, nearly all the components of the ribosome can be pulled down with the B2 sign probes. I will briefly say that we were able to confirm and validate both of these interactions, and we were able to show that the interaction of GI signs with nucleolin requires the C-terminal GAR domain of nucleolin, which is the domain which is responsible for nucleolin transport to axons. It's the domain that actually enables nucleolin to bind to kinesins. We were able to use proximity ligation assays to show that nucleolin and ribosome interaction is enhanced upon expression of GI signs. So again, we do an intrathecal AV overexpression of B2 signs, in this case in the DRG. Wait 2 weeks, do cultures from the lumbar DRG that are overexpressing these GI signs, and then running PLA on this control versus the B2 sign overexpressor. You can see an enhanced association of nucleolin and ribosomes in the cytosol in these neurons. And concomitantly with that, we were able to look at local protein synthesis in axon tips, and also in the soma of these neurons, we see a specific effect of the B2 sign overexpression on synthesis at axon tips. It attenuates synthesis at axon tips, and this we've also seen for manipulations that perturb nucleolin. So shifting translation away from the end of the axon towards the center of the cell, and this enhances growth. So finally, we went back to Shifra Bendor in a bioinformatics unit and said, we really need a way to target these GI signs. And this is rather a tall order. We want to target 450 different RNAs, specifically without targeting the 149,500 others, which are members of the family. So she did a very, very careful look at the sequences, and she identified 2 motifs that appear to be highly conserved within the GI signs and not within the other signs. And I will remind you, this is the general sign sequence consensus logo that I showed you earlier, so you can appreciate the degree of conservation in these 2 motifs, which happen to be on the POL3 promoter sites of these GI signs. And Eitan and Shifra were able to design a set of antisense oligos to target these motifs and to test their effects in DRG neurons. And so this is the antisense oligos targeting the sign. Control, untransfected. I hope you can appreciate that here you're seeing less growth than here, and this is quantified both in terms of total neurite outgrowth and the number of neurons that extend axons in this system. So 2 messages from this side. First of all, these 450 family members are indeed behaving as a separately evolving family that's under some kind of independent selection, and it has conservation of sequence relative to other signs, and one can functionally target them in a distinct manner and see the functional response.
So about 2.5 minutes left.
Yeah, I'm on my before last slide. Thank you. So just to summarize, I've been telling you about a scanning system that SOMA uses to communicate with the tip of the axon, sending RNAs out on nucleolin, getting proteins back from local synthesis. And what I've told you today is that the retrograde component of this mechanism apparently activates through AP1 transcription factors expression of a subfamily of non-coding RNAs called the GI signs, and their export to the cytoplasm can, on the one hand, apparently perturb nucleolin interaction with kinesin and thereby interrupt this mechanism, and that would be the negative feedback loop that we were looking for. But the other thing it can do is that it can interact with the protein synthesis mechanism, apparently by bringing nucleolin and its associated RNAs together with ribosomes to perhaps enhance or give a higher probability for translation of nucleolin cargo RNAs. And again, the only reason we discovered this was by setting up a hypothesis that turned out to be completely wrong, but forced us to look very closely at something that many other people have looked at and not seen before, simply because nobody paid attention to these bizarre non-coding RNAs in this type of system. And finally, I would just like to point out that the system I've described to you today is based on AP1 transcription as an input, and it affects translation as an output, you can imagine many other physiological mechanisms that would be modulated by this type of input-output, and we expect the GI signs and their analogs in other species are probably involved in regulating many different aspects of physiology and not just neuronal growth. And finally, I've tried to mention the people that went along, but these are the main credits for the unpublished story I've told you today. Christine came up with a beautiful idea that turned out to be wrong, but put us on the track to a very interesting discovery made by Indrek and being finalized by Eitan. And we've benefited from great collaborations with Jeff Twiss and his people, Zhigang He's lab, Mark Trushinsky's lab, Ricky Kawaguchi and colleagues at UCLA, Al Burlingham and Juan Alces at UCSF. And thank you very much for your kind attention.
Okay, so we can switch over to the questions. So just remind people, put your questions in the chat. So to start off with, so have you also looked if injury actually causes changes in the transposition, or where these sign elements are actually localizing in the cell?
No, we've not looked at genomic events per se.
Okay.
But I mean that the timing of that, the timing of such events would be very different, I think.
So then the next question, so if the mechanism of sign interaction with nucleolin is similar to line elements, are there similar motifs that are promoting these interactions?
I don't think so. We have tried to identify the motif in the sign, which is modeling this system. One of the dimensions we've found that the guard domain in nucleolin is required for sign binding. We have a separate set of oligos which we've generated, basically going 20 bases step by step throughout the sign sequence to see what will perturb sign binding to the guard domain of nucleolin, and we have an initial map which suggests the C terminal, the 5' end of the, the 3' end of the sign might be involved. Whether that's similar to line elements, I'm not sure yet, because we're still trying to verify that we really nail down that aspect of the interaction.
So then, does injury actually change the interaction of B2 sign, nucleolin, and ribosomes? And if there's any evidence that these signs are actually being translated?
Axons
or injury response?
Well, the signs are not translated, they're non-coding.
Yeah.
They're transcribed. So I showed you very briefly, I ran through that when we have sign overexpression, there's enhancement of nucleolin ribosome interaction, and we know that there's enhancement of nucleolin ribosome interaction upon injury. We're currently trying to validate that using the ASOs, that we can also see a change in nucleolin sign interaction, which would sort of round out the question you're asking. And we think that's the case, but this is data we still need to finalize.
Yeah. So how do you think this mechanisms is placed in times during axonal regeneration? So from what your model would say, that this is sort of a later response, so after the initial injury.
So you will start to get sign upregulation as a retrograde injury signal comes back, and that will happen in a matter of a few hours. If you talking about a rodent peripheral nerve injury, like a static nerve injury, 3, 4, 6 hours. And from then on, the question is how the feedback will change the system. And that we don't know yet. Since you're introducing a negative feedback that will then switch off the system, and then we don't know how it switches on again. So the question is it a periodic oscillator, which is what we think is happening in growth, or whether it's a one-time signal and an injury context is something that we don't know yet. And would certainly be interesting to find out.
Yeah. So then actually one from one of your fellow speakers. So is the inaccessibility of the GI sign encoding regions of the CNS neurons the only factor involved in this? And do you think it could actually be overcome?
Good question. It can be overcome in the sense that when you overexpress GI signs in these neurons, then they respond, they grow. How one would actually get an endogenous opening of chromatin in those areas, upregulation, that's an interesting question, and I unfortunately have not the foggiest idea how to even start to think about that. Hopefully smarter than us will be looking at those aspects.
So I think I have kind of a question off of that, because you did see that when you express these in other neuronal cell types, that they didn't have an effect per se, or that they didn't have that same upregulation, but they could have an effect. They could promote the axonal outgrowth. So do you think
see that although there are some concerns
about every cell type or neuronal cell type would have their own set of sign elements or these non-coding elements that are sort of based off of what can be transcribed?
Well, others have shown that B2 signs are upregulated and involved in some types of neurodegenerative disease paradigms. I'm blanking on the name of the lab. I apologize if he's on the call. It's a Greek name, and I think he works in Canada. 2 beautiful papers on signs in a neurodegeneration context. As to what happens in other physiological contexts, we'd love to know, but we came upon this not expecting to find this. We'd be delighted if people look in their data sets. The NCBI has this thing called a repeat masker. So they helpfully hide this information from you as the default if you're looking at your genomic data in the genome browser. And you need to actually click away this repeat masker to see these repeats coming up in your data, because somebody thought that it's not worthwhile for people to look at junk DNA. So I'm sure that it's involved in many other systems, and I'd be delighted if people will look.
Okay, well, I think it was a beautiful talk, Mike, and very nice discussion. And we'll return to some more questions at the very end. And now I will hand the mic over to Sun to introduce Yingxi Lin.
Yes, hi everyone. My name is Sun, and my lab is in Dendrite and Aarhus University in Denmark. And right now it's my pleasure to introduce our next speaker, who's Dr. Yingxi Lin, who graduated from Tsinghua University in China in 1991 with a degree in physics, and subsequently did her PhD with Gerhard Wagner at Harvard on the NMR studies of transcription regulation. So as you can see, she wasn't educated as a neuroscientist, but then she joined Michael Greenberg's lab as a postdoc. Michael Greenberg is one of the Brain Prize laureate in 2023. Now, quoting Michael, Yingxi took a big leap into neuroscience as a postdoc and helped bring us into the genomic era by developing gene array technology in the lab when this approach was just starting. And during her time in the Greenberg lab, she discovered an important immediate early gene called MPAS4, which is turned on specifically by calcium influx in neurons, now making this a very unique immediate early gene. And Yingxi started her independent lab at MIT in 2008, and 10 years later moved her lab to the State University of New York. And I think last year she moved again to the University of Texas Southwestern Medical Center, where she now serves as the chief.
And to this end, we generate
neuroscience research division. Now, her lab's work has revealed MPAS4 as an important transcription factor for regulating synaptic plasticity, which underlies learning and memory in the hippocampus. And along these discoveries, Yingxi and her team also invented elegant methods to identify and manipulate neurons that encode memory. Now, quoting Michael Greenberg again, Yingxi's work on MPAS4 is widely recognized and has ignited a field of study where many neuroscience labs around the world are using reagents her lab generated to investigate the function of this unique IEG in learning, memory, and behavior. Because of her contribution to science, Yingxi has received many prestigious awards, such as the Whitehall Foundation Award and the NASAD Independent Investigator Grant, and she has served as a regular member of the NIH study sections and has taken up leadership in graduate student education. Yingxi's intellectual path is testament, I think, to the fact that the field of brain research is indeed a melting pot of multidisciplinary background, talents, ideas, and techniques. And it's also relatable for me personally, because my switch to neuroscience also happened during my postdoc when I joined Erin's lab. And with that, I very much look forward to your talk, Yingxi. You're muted.
I'm muted.
Now we can hear
Now
you.
we can hear you.
Yeah.
Now we can
alright.
hear you. Just share your screen again. Yeah.
Yeah.
Super.
Thank you, Chow, for the kind introduction. You can see my screens now, right?
Yes, we can.
All right. Yes. So I'd like to begin by congratulating Erin, Christine, and Mike for winning the brand prize. And I have the distinct honor to receive my postdoc training in Dr. Mike Greenberg's lab. And as Chow mentioned, that I wandered into neuroscience without knowing much of neuroscience. And in Mike's lab, this is really where I learned everything about neuroscience and started my career in neuroscience. So now you see, even though you see Mike holding a pipette here, and for the record, I've never seen him doing any experiment in the lab. But I did have the privilege to witness Mike as a brilliant scientist and a great mentor. So what attracted me to Mike Greenberg's lab is this fascinating cellular process called neuronal activity regulated transcription. And when neurons are activated and receiving signals from synapses, a lot of local events happening at the synapses, including local protein synthesis, the process that Erin and Christine have been studying. And however, there's a very critical event that's happening that transmit the signals received at the synapses into the nucleus to turn on gene expression. This is called activity regulated gene transcription. And this is one of the ways the cells try to register what it receives. And showing here as an example is the activation of CFOS in its original publication from Mike. And CFOS probably is the best known activity regulated gene and has been used to label behaviorally activated neuronal populations. So what I decided to do today in honor of the topic that Mike won the Brand Prize for is to share with you some evidence that my lab collected over the years, showing that neuronal activity regulation gene transcription programs encodes cellular memories that constitute memory engram. So the neuronal population that in which the activity regulated transcription programs are activated are referred to as active neuronal ensembles here in my talk. So within the neuronal activity regulated gene programs, it's a group of genes called immediate early genes or IEGs. These genes are activated very rapidly within minutes, and they are also activated very robustly. And you can see some familiar faces here like FOS, ARC, and EGR1. And many of the majority of these IEGs are also transcription factors, so they turn on the expression of downstream targets to modulate neural circuits in a variety of different ways. So in most cases, these IEGs are used as activity markers indiscriminately, but here I want to draw your attention to the fact that each IEG transcription pathways can lead to very distinct downstream, can modify neuronal circuits in very distinct ways. In other words, each transcription pathway can encode very distinct cellular memories. And for example, I want to compare two of them. FOS and MPAS4, as Charles mentioned, is a IEG that I identified when I was a postdoc in Mike's lab and continue to study in my lab. And FOS is known to regulate neuronal excitability and is required for LTP. You can see it functions at this spectrum, at this end of the spectrum. On the other hand, MPAS4 seems to function in a very opposite way. MPAS4 is known to work for my lab, and many others have shown that MPAS4 regulates inhibitory synapses onto excitatory neurons. So it functions at this end of the spectrum. In fact, many years ago, my graduate student, Kartik, also found that FOS and MPAS4 can activate it in they are activated in not always in the same population of neurons. What you see here is that within the hippocampus, after contextual field conditioning, and MPAS4 activation is highly concentrated in CA3 and a little bit in the dentate gyrus, which you cannot see very clearly here. And this is very different when you look at FOS. FOS induction is throughout the whole hippocampus. So these early observations led us to form the hypothesis that information encoding is encoded by functionally distinct neuronal ensembles that can be defined by different activity-dependent transcription pathways. So there is a molecular sorting event here that sort the incoming information into different neuronal ensembles to encode different cellular memories. So to really prove this hypothesis, my former postdoc, Andreas, and teamed up with a very talented research assistant, Yoni, to develop a system we call Robust Activity Marking System, or RAM system. In the heart of the RAM system is a RAM promoter that the promoter can be activated by the binding of one of the IEG transcription factors to drive the expression of reporter molecules within the activated neuronal population. So I won't go into the details that the RAM system also has a temporal control that prevents the reporter molecules from expressing until it is within the desired experimental window. So now by designing different RAM promoters, we'll be able to capture neuronal populations defined by active neuronal ensembles defined by different activity-dependent transcription pathways. And by expressing different type of in reporter molecules, you can use the RAM system to label the ensemble neurons or to manipulate their functions using chemogenetics and optogenetics, or to monitor their activity in vivo in live behaving animals using calcium imaging. And the RAM system is purely, it's an AAV-based system. It's very compact, and most of the components can be packed into one single AAV, so it can be easily applied to species other than the mouse. So I want to show you an example where you see how the active ensembles look like in the hippocampus. In this one example is in the dentate gyrus. And what you see here is the RAM virus is injected into the dentate gyrus, and we train mouse using in the contextual field conditioning. And the RAN, in this case, the RAN reporter is the reporter drives the expression of a nuclear localized red fluorescent protein called MK2. So you can see here, consistent with the sparse coding in the dentate gyrus, the population of active neuron ensemble captured by the RAN system is about 1 to 2% of the dentate granule cells, and which is significantly higher than the population you see in the home cage.
On that, we took.
With this RAN system established, we were ready to test our hypothesis that the contextual field memory is encoded by this functionally distinct active neuronal ensembles. So my very talented former graduate student Xiao Chen teamed up with Max and Xiao Zhen and Mei Zhen, who's a current graduate student. They decided to test our hypothesis in the dentate gyrus. So dentate gyrus is a region is known to be involved in contextual memory discrimination. So they designed a very simple memory discrimination assay that they train animals in with the conventional contextual field conditioning in context A, and then after 24 and 48 hours, mice is in the condition context or very similar context.
Yingxi, your voice went to a lower volume. Could hardly hear you right now.
Can you hear me?
Oh, we can hear you, but just barely.
Very quietly, though. Can you speak louder or?
Okay.
You're
Okay,
back. Yes.
Is it better now?
Yes. Yes. Good.
Okay. Sorry. I don't know how much you missed. And so so with this assay, they tested whether the animals
Binders were
distinguish three different contexts: context A, which is the condition context, and context B, which is a very similar context, and context C is used as a control, as a neutral context. As you can see, the mice can really tell these three contexts apart, and they freeze at a high level in context A, and at the basal level in the context C, which is considered a neutral context. However, in context B, they can tell it's very similar to context A. They freeze at a high level, but the freezing level in context B is always significantly lower than that in context A, because they could tell context B is not identical to context A. So the freezing level can be plotted out as a function as a similarity between the conditioned context and the memory retrieval context. So this curve is very similar to the discrimination curve seen in human studies. So what we hope to find and define is that some of the neuronal ensembles will push the curve to the right to enhance discrimination, and others will push the curve to the left to promote generalization. And it turns out the neuronal ensemble defined by MPAS4 is required for memory discrimination. So here I'm color-coded the MPAS4 population in blue, and sometimes I also call them NRAN ensemble neurons because they are labeled by MPAS4 dependent RAN reporter. And on the other hand, the FOSS ensemble, we found color-coded in red, we found is promoting memory generation. So now I'm going to show you some key experiments from this study. So what we did is to manipulate the activity of the FOSS and MPAS4 ensembles during memory retrieval. and see whether this manipulation will affect the animal's ability to distinguish different contexts. When we chemogenetically inhibit the force-dependent ensemble, what you can see here, the discrimination between A and B is significantly improved, which is, you can see here based on the freezing levels and also from the discrimination index calculated using this formulation. And this result suggests that normally that typically force ensemble is involved in promoting memory generalization. On the other hand, when the MPAS4 ensemble is inhibited, the discrimination between A and B is completely abolished, suggesting that the MPAS4 population is required for memory discrimination. Consistent with the fact that the dentate gyrus is required for discrimination between between very similar contexts, but not required for very distinct contexts, the discrimination between A and C is not abolished by this, it's not affected by this manipulation. So we next wanted to understand what type of cellular memories are encoded in this two ensembles to allow them to have such distinct functions. And so it turns out that the fast ensemble neurons, they after learning, they selectively recruit excitatory input from medial entorhinal cortex, which is one of the three major excitatory inputs received by each granule cells. So in this experiment, Tsien conducted a dual patch experiment by patching on a pair of neurons. One is Fren positive, the other is Fren negative, and then and record the intensity of postsynaptic current this pair of neurons received by activating one of this each three of the excitatory inputs. As you can see here, the false ensemble neurons receive much stronger excitatory input from MEC. So this result suggests, it's very exciting, suggests that the input from MEC onto the false ensemble neurons may be the driving force for memory generalization. So indeed, when Tsien inhibited the MEC, what he saw is the memory discrimination between context A and context B is dramatically improved, exactly like what we saw when we inhibited the false ensemble neurons. Inhibition of LEC has no effect on
Here, and this is quantified both in.
So how about the MPAS4 population of neurons? And very remarkably, among the many different types of GABAergic inhibitory neurons, the MPAS4 ensemble neurons selectively recruit inhibition from CCK expressing GABAergic interneurons. So again, Tsien using this dual patch experiment to measure the evoked inhibitory postsynaptic currents received by this pair of neurons by optogenetically activating different subtypes of GABAergic interneurons, and the NREN neurons receive much stronger inhibitory.
Yeah, I'm on my
interneurons. So this result is really exciting to us because this suggests that CCK interneurons might be involved in memory discrimination, which had never been shown before. So because CCK is also expressed in excitatory neurons. So we used a genetic intersection method to selectively target CCK expressing GABAergic interneurons. So when CCK expressing GABAergic interneurons is inhibited, the
mechanism apparently activates.
combination between context A and B is completely abolished, completely mimicking what we saw when we hemogenetically inhibit the MPAS4 MPAS4 dependent neuronal ensembles. When the PV or SST interneurons are inhibited, the memory discrimination is not affected. So this slide is to summarize what I have told you so far. That what I show you so far suggests that the FOS and MPAS4 dependent transcription pathways encode very distinct cellular memories in very distinct active neuronal ensembles. The cellular
and its associated RNA
is the learning-induced synaptic change. So learning selectively strengthens the input from MEC onto FOS-dependent active neuronal ensemble neurons to drive memory generalization. And on the other hand, learning selectively recruits inhibition from CC
by setting up the hypothesis
interneurons to regulate memory discrimination. So we know this cellular memories encoded on the FOS and MPAS4 dependent neuronal ensembles are actually encoded by FOS and MPAS4 dependent transcription pathways, because when we artificially activate the expression of FOS and MPAS4 in a sparse random population of neurons, and these neurons acquire similar synaptic properties as the FOS and MPAS4 active neuronal ensembles. As you can see here, neurons overexpressing FOS recruits more excitatory inputs, and neurons overexpressing MPAS4 selectively recruits inhibitory inputs. And what's even more remarkable is when, in the case of MPAS4, expressing MPAS4 alone actually can selectively recruit CCK inhibition. So my student Mei Zhen did the dual patch experiments and recording the inhibitory postsynaptic currents on a pair of neurons. And one is overexpressing MPAS4, and the other is the control neurons. And when you see here, the neurons overexpressing MPAS4 receives significant stronger inhibition from CCK interneurons, and but not from other GABAergic interneurons. So next, we also wanted to know whether the cellular memories are actually encoded by learning experience. So in order to show this, we train animals with different strengths of CFC protocol by using different intensities of food shocks. And as you can see, animals receiving stronger food shocks freeze at higher levels, suggesting that stronger learning leads to stronger memory. And what we found is that the stronger memory correlates with stronger synaptic changes that you see on both the FOS dependent ensembles and also on the mPAS4 dependent ensembles. So these results suggest that the cellular memories correlates with the strength of correlates with the strength of the memory, and might be the cellular memories might be regulating the output of the memory. So last, we wanted to see whether these distinct cellular memories translate into different in vivo functions of this neuronal. Yeah, so so we decided to use fiber photometry to monitor the activity of the FOS and mPAS4 ensembles in vivo during memory recall. So we targeted the GCAMP expression into the FOS and mPAS4 ensemble neurons and monitored the activity while animal performing the memory discrimination assay. So by measuring the average calcium activity during memory recall in relative to the activity at the home cage, and what you can see here, the FOS ensemble neurons show very strong activity in both context A and context B. But what's very surprising is that, very interesting, is that the mPAS4 ensemble shows significantly higher activity in context B than in the condition context A. And this is pretty unusual. And what's even more remarkable is that the activity level, the difference in the activity level in A and B in the mPAS4 ensemble neurons correlates with the ability with the animals correlates with the animal's ability to discriminate between context A and B, which is not true in for the FOS ensemble neurons. So in summary, today I show you that neuronal activity regulated gene transcription programs encode the cellular memories that constitute the memory engram. And the memory engram is not homogeneous. It consists of very distinct cellular memories that are that's defined by different distinct activity encoded by distinct activity-dependent transcription pathways. And although in my talk, I mainly focused on learning-induced synaptic changes as cellular memories, the cellular memories encompass many other different components, such as genomic and epigenomic changes, which we still don't have a very full picture of. And of course, future work is needed to understand how cellular memories are distributed to achieve optimal functional outcomes. So with that, I want to thank the people who contributed the work. So this work is mainly conducted by a very talented graduate student, Xiao Chen, who's now as a postdoc at Stanford. And also Mei Zhen, my current student, also contributed significantly to the work, and our collaborators along the way. And the RAN system is it's very instrumental for our research. So the project is spearheaded by a very talented former postdoc, Andreas, who's now a associate professor in University of Copenhagen, and Yoni just graduated, got his MD PhD degree and started his fellowship at Stanford, and our funding agent. And also I'd like to thank all the former and current LINUP members, and it's a privilege to work with groups of many very talented and dedicated people in my lab. As Erin mentioned, that we just moved to UT Southwestern, and I hope you interested students and postdocs will join us there in Dallas. Thank you very much.
Thank you for the beautiful talk, Yingxi. So we have a few questions. First one from Dick Tsien, who asked, who said, very nice talk. Are the intrinsic spiking properties of the false or NPAS defined GC subsets unchanged? So only synaptic changes are required.
So can you repeat the question?
The question is whether the intrinsic spiking properties of the false or NPAS4 subsets unchanged. Are the spiking properties changed or not between the two kinds of neurons?
So the question is whether the spiking properties of the neurons are changed. So this is a question we don't know. So we are in the process of doing, because the fiber photometry doesn't have the cellular resolution. So we're in the process of to address this question. So I don't know whether also the question refers to whether they are intrinsically different, those two populations. I don't know what the question referred to, whether it was after or before.
That was actually one of my kind of naive questions. Are there, I mean, in another way, I asked, like, does any neuron express both of them? If
Yes.
you don't, then they're, yeah.
Yeah, so actually, there are multiple levels of controls, and also in some brain regions, we see more distinct distribution than others. So there's no reason why these two pathways can overlap. And just in the case of DG, this in this particular setting, they are distinct, and it's a place. It's very very convenient for us to test our hypothesis, but they are not always overlap. That's the main message. But in cases they can be overlapped, and they can, in the same neuron, they can modulate both excitatory inputs and inhibitory inputs, and also in some cases, MPAS4 can also modulate excitatory inputs as well, as we shown in CA3.
So we have another question from the audience named Ilaria Barona, who said very beautiful work. And she asked a question in the context of the circadian clock. She asked whether you have you want to try this paradigm in daytime, and whether you think the segregation between the CFOS and MPAS could be the mechanism to consolidate information during the daytime.
That's a great question, because that I will, well, I don't know whether they are here. I will refer this question to my new colleague at UT Southwestern, Joe Takahashi, and Joe Takahashi's lab actually is also studying MPAS4 in circadian regulation. So I actually don't know the answer, and maybe they will be able to answer this question. But I think this is a brain region that hasn't been looked at very much, and I think I'm excited to see what they find.
Okay, so let's take one last question from Mike, and who said very intriguing. Can neurons alternate expression of CFOS and PAS4? They regulate each other.
Yes, I think Mike asked a very interesting question. So in fact, you see MPAS4 and FOS bind onto each other's promoters. And we know in some cases, actually, we also saw in one of our early studies, not always, in some cases, MPAS4 can regulate FOS expression. And I think vice versa, although we haven't seen it, but I wouldn't be surprised that FOS can also regulate MPAS4 expression. So in a way, I think there are all these different IEG pathways, and we sort of conveniently picked FOS and
Sure.
they seem to be functioning at the very opposite end of the spectrum. And so like every system, there's always a balance, right? There's a yin and yang, there's just like memory discrimination, there's memory generalization, there's always a balance. I think it's sort of conceivable these two pathways can auto-regulate each other to achieve certain type of systems and cellular balance.
Thank you.
Thank you very much, Yingxi,
Yeah.
for your talk.
Yeah, thank you, Yingxi. That was really beautiful.
Thank you.
Okay, so I will now introduce our last speaker, Dick Tsien, who is one of my scientific heroes. Dick did his undergrad degree in electrical engineering at MIT. He got both his bachelor's and his master's there. He then was a Rhodes Scholar and got his DPhil degree in biophysics at Oxford, where he worked with Dennis Noble. Dick's first faculty position was as an assistant professor at Yale University in the physiology department, and there he moved through the ranks to become a full professor. And then in 1988, he became the founding chairman of a brand new department at Stanford called Molecular and Cellular Physiology. And there he crafted a state-of-the-art neuroscience department focusing on, well, not just neuroscience, but focusing on interesting aspects of neurons and focusing in particular on the cell biology of neurons and other cell types. And that's where I met Dick when I was a postdoc. And I have to say that the department that he created was a fabulous environment, and for me represents like the golden years of my life as a scientist, the times I spent in Dick's department there. So he was at Stanford for over 20 years, and then in 2011 he became the director of the NYU Medical Center's Neuroscience Institute and also the director of the neuroscience program there. And there he served for over 20 years. He still has his lab there, and he recently stepped down as director. Dick has gotten many, many prizes and accolades. For example, the Axelrod Prize from the Society for Neuroscience, the Gerard Prize. He's a member of the American Academy of Arts and Sciences, the National Academy of Medicine, and the National Academy of Sciences. Dick's legacy, scientific legacy, is vast. He began interested in ion channel physiology, made huge contributions to the molecular diversity and functions of calcium channels, which he still works on today, in particular dissecting the various types of calcium channels that give rise to different patterns of neurotransmitter release. He made very important contributions to the field of long-term potentiation and homeostatic plasticity. And more recently, and he will talk about this, I would say, in this long legacy, is how activity in neurons is linked to intra and extracellular signaling. Now, not only does Dick have his important scientific legacy, but he also has an amazing legacy of people that he's trained. And it's a long list of many esteemed neuroscientists, people like Steve Siegelbaum, Bruce Bean, Diane Lipscomb, Robert Malinow. Of course, I have to mention also Dan Madison, who was my postdoctoral advisor, and give a shout out to my two homies, Felix Schweitzer and Antonio Malgaroli, who were my contemporaries. Anyway, Dick has made huge scientific contributions, but also huge contributions to the people that he's trained, and also the people that have been in the departments that he's been chair of. So it's one of those things where it's like 6 degrees of separation with Dick. And I would bet that almost any neuroscientist you meet, it would be more like 3 degrees of separation before you'll find someone who's worked with Dick or has been in one of their departments. And so it's a great pleasure for us to have you as our closing speaker, Dick, and I very much look forward to your talk.
Thank you, Erin. That was very generous. And now the part of the talk where Martin Mayer sits on the edge of his seat. hoping that we can actually get things to work. I'm hoping that you can see my title slide
We can.
without, you cannot?
We can, yes.
Okay. So Mike Fainzilber and Yingxi Lin gave really interesting talks. And I'd like to round out this morning by thanking Erin, Christine, and Mike Greenberg for their pioneering contributions to the field. They really deserved the prize, and everything that was kindly said about me could be said about them. I'd like to change gears a little bit and talk about unpublished work for the most part. Think of it as kind of a nano symposium at SFN, only with a few tweaks to the slides. So the Lundbeck Foundation was instrumental in bringing us together. And Martin Meyer, who is also a postdoc working with Stephen Smith at Stanford at the time, is an old colleague. And I thank him for basically holding our hands through all of the media aspects of this and the teams aspect. Really, science is about teams. And I'm really grateful to the opportunity to speak to you. Thank NIH and also the Vulnerable Brain Project for their support. Yingxi did this very nicely, probably nicer than I would, but let's say that excitation transcription coupling is one part of the cell biology of learning and memory. Messages get from synapses or spines or cell bodies all the way to the nucleus, where the vast information storehouse in our genomes is tapped. And that in turn leads to the transport of macromolecules, whether they be proteins or messenger RNAs, back out to the synapse, where synaptic strength changes are consolidated and reinforced and made to live on. And that's how Mike Fainzilber can remember the anecdotes that he told us as we were getting together. And there are a lot of questions, and I'm not going to try to deal with them all, but there are questions for molecular scientists about which molecules initiate the signaling, where in the cell does the signal come from, how is the downstream message conveyed to the nucleus, and what are the functional consequences for behavior and disease. You'll be hearing that more about that in future webinars. And what is the relationship to long-lived synaptic plasticity? I won't try to do justice to all the active people working in the field. Huda Khaled, Juan Ma, and Xiaohan Wang just published a review in Nature Reviews Neuroscience that provides a long reading list for anybody interested in this field. Instead, I'd like to focus on the idea that the ion channels that support excitation transcription coupling are beautifully localized in the spines that decorate the dendrites of excitatory neurons. Here you can see that, for example, in work from DiBiase from Austria, showing that co-localization of green fluorescent protein labeling the spine head and a HA antibody, an HA-tagged calcium channel, show excellent co-localization. The yellow spots are spines sticking out of the plane of the section, indicating that almost all the spines have L-type channels. So even though many people cartoon L-type channels as being in the cell body, they're actually in the dendritic spines themselves, and where they interact in their voltage dependence and their delivery of calcium with glutamate activated channels. So when you look at the expression of a nuclear transcription factor and its phosphorylation and activation, here in a paper by Sam Cohen, you can see that 50 Hertz extracellular stimulation produces a nice increase in phospho-Krebs, which can be blocked either by blocking glutamate receptors or by blocking L-type channels. And if you block both of them, you get hardly any greater effect than if you block either one alone, indicating some sort of sequential process where both of them signal in series with each other. This type of arrangement of co-involvement has also been seen in spike timing dependent plasticity from Bi and Pu, showing the effect of nimodipine blocking the potentiation. In work from Anjali Yaksha's lab with the Cav1.2 knockout, which also shows a reduction in something that Erin would love, theta burst induced synaptic potentiation. And so you can see both an early effect and a late effect. And finally, in brilliant work from Bittner and McGee, the induction of a plateau in the intracellular recorded potential from a mouse navigating a maze is blocked by nimodipine, an in vivo experiment showing you the involvement of LTP in the first stages of one-shot learning and memory. So how are the roles of Cav1, in other words, L-type channels, in early synaptic plasticity, LLTP, and ET coupling related to each other? Well, one way of approaching this question is to ask, where in the cell does depolarization need to occur in order for signaling to the nucleus to arise? This would be a question that Mike Greenberg would love if he were here, because he began as a postdoc with Ed Ziff, depolarizing cells that were made into neurons by treating PC12 cells with nerve growth factor. Nowadays, we know the molecular structure of the L-type calcium channel in exquisite detail in work from Niang Yan's lab. And you can even see a dihydropyridine molecule located at the center of that structure. The dihydropyridine molecule has an essential NH group, and the NH group sits among the helices of the channel, where the NH part can interact with a key residue defined by Adachi Akahani's group that in turn governs the inhibition of the channel. So by irradiating a channel that contains a 1,4-dihydropyridine, either nifedipine or nimodipine, you can convert the NH group into simply an N group. And that removes the interaction with the key serine residue and prevents the inhibitory effect on the channel. Now, a excellent MD PhD student, Nathaniel Mandelberg, saw this possibility and realized that he could locally unblock Cav1 channels by light and therefore initiate ET coupling at a location of his choice within the neuron. And so here you see the results of irradiating with a square a box of light. And if you monitor the calcium response to a potassium depolarization in the irradiated region, you find that there is a nice big calcium response. Whereas in the remaining region, in the off-target region, where the dihydropyridine has not been photo switched off, you see a very small response. That small response is due to the other types of channels that Erin referred to, the Cav2 channels, N, PQ, and R-type channels. But if you apply a very expensive boatload of toxins, you can inhibit that remaining response to show that it is due to channels that are not involved in ET coupling. Instead, you can see that the irradiation, that only with treatment with nimodipine, the irradiation restores the calcium transient pretty much to what it was in the absence of drug. So now Nathaniel can go in and use light to pierce a hole in the clouds and therefore allow light to shine in, or allow a signal to transfer, and that signal can take place all the way to the nucleus. So this shows group data showing the efficacy of the laser in restoring the area under the curve of these calcium transients. And this shows the experiment that you're probably most interested in, and that is what happens if the target region is a dendritic spine. Here you can see that the targeted spine shows a nice calcium response, whereas a neighboring spine in an off-target region shows no response. So that means we can activate calcium entry at will at a particular location. Once again, group data, once again showing the efficacy of the light in restoring the calcium response. Now, this shows a movie that Nathaniel took, indicating, and I want you to look at this region in purple very carefully and watch it, and the signal that you're looking for occurs about halfway through the movie. You can see the calcium response selectively localized to the irradiated spine and not to the other one. So now Nathaniel found a way to keep track of the neurons that were irradiated and sometimes only in a particular region, and then used the standard kind of Krebs phosphorylation assay using an antibody. This shows irradiation of the cell body and indicating that the target cell has a nice Krebs response in contrast to a cell that was not targeted with light. This is a very consistent finding. There is no response if you have only nimodipine and don't depolarize, or if you have nimodipine and you do depolarize, or if you or shine the light in a different area. It's only if you shine the light on the cell of choice that you see the restoration of the Krebs response. And this also holds for dendritic irradiation. You can see a very nice response in the cell body with the activation of only a small number of spines, somewhere between 3 and 30. That produces a good restoration of the Krebs response, indicating that we can, number one, optically pinpoint the source of signaling for ET coupling, and number two, that a small number of spines is sufficient if stimulated for 3 minutes to send the signal all the way to the nucleus that activates phospho-Krebs. So the calcium channels can signal either from the soma or from the dendrite, even though the bulk of the calcium channels in the cell are inhibited. Now, why is L-type channel advantageous for cell biology and for learning and memory? One obvious possibility is that they could provide calcium influx synergistic with NMDA receptors, and perhaps in sequence with NMDA receptors, where the NMDA receptors depolarize the cell and activate the L-type channels. They're also good at responding to spine depolarization. So in experiments studying BTSP and spine properties, Harnett, McGee, and Bittner and their colleagues, Christine Greenberger, depolarize spines either physiologically or optically, and either way, you can get a signal as a result of L-type channels. Another possibility studied by Mark Dell'Acqua is that you can propagate an action potential to the soma. That's certainly important for the activation of a transcription factor called NFAT. There can be close communication with the ER. Many people have studied this, including Jennifer Lippincott-Schwartz, who shows that the ER is very periodic and sticks out branches that interact with L-type channels. Finally, the L-type channel is very good at binding CAMK2, and this we find is important in signaling to Krebs phosphorylation. That's the work of Juan Ma. But in this talk, I'm going to focus on the idea that the L-type channel is actually a voltage sensor that can do without its calcium flexing properties. It can act as a monitor of voltage, whether that be in the dendrite, in the shaft, or in the spine head itself. And this is important in sending a conformational signal that is activating nuclear transcription. So how does this work? Well, in schematic form, the L-type channel has charged S4 regions schematized by these plus signs, and that conformational signal works to activate not only gene expression, but also to cause the anchoring of CAMK2. You can study this by blocking the flux through the L-type channel with cadmium at a concentration which completely eliminates the flux through it. And you still see a role of conformational signaling envisioned, for example, by producing calcium entry through a different channel to replace the calcium entry that the L-type channel would have normally induced. And that creates successful signaling to the nucleus, gene expression, phospho-Krebs. If you then want to toggle the system, we can use nimodipine, which drives the channel into a closed state where the S4 regions are in the down state, and the channel not only cannot supply flux, but it cannot supply its conformational signaling. And when that happens, the signaling to the nucleus is abolished. This is work of Mike Tadros, Boxing Li, and myself, and also was presaged by some very insightful experiments by Daphne Atlas. So how does this channel then interact with endogenous calcium sources? What we think goes on is that the NMDA receptor and the L-type calcium channel work as a team. The NMDA receptor is much better at supporting calcium flux, but it doesn't undergo a very large voltage, doesn't have a large voltage dependence. Instead, the L-type channel confers a very steep voltage dependence that responds to stimuli like dendritic plateaus. And this combination is what's important in driving the signaling. And you can see this experimentally by directly applying NMDA to selectively activate the NMDA receptors and looking at PSD95 regions for whether there's a local CAMK2 signal. If you apply NMDA with cadmium present to eliminate the flux through the L-type channel, you nonetheless see a big increase in the accumulation of phospho-CAMK2 at those dendritic spines, as if it were getting ready to either support signaling to the nucleus or local LTP. If you apply nimodipine, which doesn't affect the flux because the channel is already plugged with cadmium, then you see a great reduction in the amount of phospho-CAMK2. So this is suggestive of immobilization by the NMDA receptor, but this result, the much lowered reduction of phospho-CAMK2, suggests that somehow the L-type channel is getting into the act. This is almost down to baseline, and if you average over various regions of interest, you find on average that it's just slightly above baseline. Sometimes there's a phospho-CAMK2 signal, sometimes there's not. So what are the spatial temporal properties of this cooperation? Now, Nathaniel and Huda perform an experiment that goes all the way back to Carl Dijsselroth when he was a postdoc along with Erin and Martin. This is an experiment comparing the behavior of calcium provided by the NMDA receptor with two different calcium buffers, with the EGTA, which allows the calcium to spread about a micron, or BAPTA, which, as my brother Roger showed, is good at supporting calcium flux over a much narrower region. And now we're going to look at Krebs as the final outcome and compare the results of delivery of BAPTA or EGTA. And what we see is that if we use the NMDA receptor as the calcium source, we find local signaling close to the NMDA receptor, which is blocked by BAPTA, but not by EGTA, as if to say the signaling must be all very local, all within the dendritic spine, almost as if we were using a site elegantly defined by Xiaohan Wang when he was a postdoc in Roger Colbrand's lab, namely to show that a particular binding site for CAMK2 in the N-terminal region of the L-type channel, either CAV 1.3 or CAV 1.2, is responsible for binding to CAMK2 and provides a very stable anchor for the kinase. So the signal is present if we have a wild type or a selected L-type channel, but it's abolished if we modify just 3 residues in the CAMK2 binding site. So now our scenario involves a transient interaction between the NMDA receptor and CAMK2. The NMDA receptor has a particular site, serine 1303, which is important for CAMK2 binding. A residue, this residue is a phosphorylation target of CAMK2, as shown by Mary Kennedy. And when that residue is phosphorylated, the affinity of the NMDA receptor for CAMK2 is actually lowered. This seems initially paradoxical. If you're trying to get CAMK2 to stay in the spine, why lower the affinity for its favored target? Well, here's a scenario that puts all of that together. The NMDA receptor provides calcium flux, and that activates CAMK2, which is normally localized on F-actin cables, out of the way and not ready to support signaling to the nucleus. But when it sees calcium, the CAMK2 changes its conformation and is liberated and is allowed to diffuse to the NMDA receptor, where it binds with relatively high affinity. However, the activation of L-type channels causes a alternative trapping site, and perhaps there is a handoff between these two, which allows the NMDA receptors to be the first target of CAMK2, but the L-type channels to be the eventual target. So Huda Khaled, an amazing student in the lab, tested this by using nimodipine as a effector of CAV1 conformation. Here we see what happens if we stimulate the spines with NMDA in the presence of cadmium to prevent L-type calcium flux. After 15 seconds or 60 seconds, there is an increase that is on average sustained in the amount of CAMK2.
So what we hope to.
However, if we repeat this experiment in the presence of deactivation of the CAV1 channel, we find that the response at 15 seconds remains, but the response at 60 seconds is almost completely abolished, indicating that somehow the CAV1 channel is having a role in this. And unless you have the conformational signal of the CAV1 channel, you don't have a permanent or lingering effect of CAMK2 in the spine. So the early dependence is on the NMDA receptor, the later dependence is on the CAV1 channel. So Huda then took advantage of the fact that there's an antibody to serine 1303 and checked to see whether serine 1303 came into the act. In control, you can see that eventually serine 1303 is heavily phosphorylated, decreasing the affinity of the NMDA receptor. But in the presence of nimodipine, that phosphorylation doesn't occur. So to wrap this up, we think that CAV1 conformational signaling is driving phosphorylation of the NMDA receptor, leading to the following scenario. The NMDA receptor provides the calcium flux that mobilizes CAMK2. CAMK2 transiently dwells with the NMDA receptor. A plateau or depolarization activates the L-type channel that then allows the L-type channel to phosphorylate, drive the phosphorylation of serine 1303 and provide an alternative target for CAMK2. So in the steady state, the L-type channel and not the NMDA receptor is the target for CAMK2. To summarize and allow Erin to close this up, optically localized recruitment of L-type channels tells us where we can trigger ET coupling. The L-type channels can open in the dendrites, and even with a blanket of nimodipine around the cell to drive signaling to the nucleus. A surprisingly small number of spines is enough to trigger ET coupling, and Nathaniel would guess 3 to 30. A voltage-dependent signal from the L-type channel synergizes with NMDA receptors to support ET coupling, and including CAMK2 translocation. The voltage signal from the voltage-dependent signal from the L-type channel is needed for the CAMK2 translocation to be persistent, of relevance to ET coupling and LTP. And finally, we know the substrate that the molecular mechanism of this, it's a site that was identified by Roger and Mary Kennedy, and we think this is what allows the handoff from NMDA receptors to L-type channels. So I'd like to acknowledge the excellent work of Huda, Nathaniel, and Shahan in generating the data, the current members of my lab, and some wonderful alumni who, through the years, have contributed to developing this field from the time that we knew that L-type channels were different than N-type channels and weren't so important in inducing synaptic transmission, to the work of Greenberg and Ziff, and also Greenberg, Kern and Morgan. And finally, this is a picture from my office window showing the snow on the Chrysler building, kind of echoing a skyscraper in our logo. Greetings from snowy New York. Thank you very much.
Thank you very much, Dick. Thanks for sharing that really cool unpublished story. So I'll start off with a question. I was impressed and surprised that the localized activation of the L-type calcium channel with just 3 to 30 spines activated was sufficient to get Krebs signal, when we also know that, depolarization is used to activate Krebs. So what does that say about the dynamic range of Krebs signaling? Is it like an on-off switch, or is it seeing shades of gray?
I think it's shades of gray. Remember that we're focusing on localization, and we're applying a 3-minute 40 millimolar potassium stimulation to make sure that we get a signal and get a nice strong signal. LTP is not made in vivo by that strong a stimulation. What is likely to happen is that the activated spines draw upon a reservoir of resources nearby, including gamma KMK2, which Juan Ma showed translocates to the nucleus, beta important, which Kelsey Martin's lab shows is important in conveying signals to the nucleus. So we're studying things which are focused on where rather than how much. I think with a physiological plateau, 3 spines might be enough to do it and might not be enough to do it. So we're not stressing the quantification. Ryohei Yusuda also showed that a small number of spines is able to signal to nuclear MAP kinase.
Okay. Let's go to the chat. Question from Chow: how abundant are postsynaptic L-type channels in comparison to the NMDA receptor to allow this handover?
That's a really good question. I would say that the L-type channels are probably less abundant, and all the more reason that they are actually biochemically toggled in their binding of KMK2. It's not a fight, a tug of war, where the number of people on either side are critical, but rather the fact that there's actually a mechanism whereby the NMDA receptor is told essentially, you got to share now. If you've got kids, you know this, and it's time for sharing. And the L-type channel and its voltage-dependent signaling is important. So having gotten the KMK2 to the postsynaptic density, then there is the possibility of a handover.
So, related to that, at the beginning, the images you showed where you were making the case that the channel is localized in the spine, those were epitope tag receptors. And so I was curious as to whether you've have similar images of endogenous receptors, maybe using expansion or super resolution microscopy to get at the localization, the relative spatial positioning of the L-type channel and the NMDA receptor.
That HA tag construct was
discrimination.
So if you like, it's an endogenous channel, not overexpressed, but okay. So we think the localization is authentic, but a good experiment that both of you folks are suggesting is to do the same HA tag strategy on the NMDA receptors, where the epitope is the same, and one would have a reasonable possibility of comparing things quantitatively and stoichiometrically. The claim here is not that the NMDA receptors and the L-type channels are comparable in calcium flux. The claim is that the L-type channels are responsive to voltage as voltage-dependent channels, and they have the benefit of a lot of charges within the S4 region of the channel, whereas the NMDA receptors magnesium dependence only has the 2 charges on magnesium and is not nearly as voltage dependent. So that nature has put these 2 molecules side by side with each other to have an AND function between ligand signal and a voltage signal. And that voltage signal is very important for certain forms of plasticity, like BTSP, and indeed for LTP, where, as we all know, it's a combination of depolarization. And it's my claim that because NMDA receptors have a voltage dependence, which is uncontrovertible, shown by Mark Mayer, for example, they have some voltage dependence, but the real voltage dependence that makes BTSP is due to the L-type channel.
Thanks, Dick. Can I ask you to stop sharing your screen so that we can see each other a little better? Next question. Very nice presentation. Can CAV1 interact as well with other calcium channels in the ER, like the IP3 receptor and ryanodine receptors?
Can you not see my screen now? I
No,
can
we
see
still
you.
see it.
Oh, okay.
And I'm worried about your meeting with Celine Santiago in 7 minutes. That's why I'm.
Okay.
Yeah. Celine's going to have to wait.
Yeah.
Okay. Sorry. How's that?
No, maybe Martin, can you make override and stop sharing? Anyway, let's move on to the question about the whether CAV1 interacts with other calcium channels in the ER, like the IP3 receptor or the ryanodine receptors.
L-type channels are known to interact with the ryanodine receptor. And among other people, Jennifer Lippincott-Schwartz and her colleagues have a beautiful paper in progress where they showed that the ER has periodic protrusions that stick out toward the L-type channel and probably make complexes with it. A lot more like skeletal and cardiac muscle than one might have imagined about a neuron. And so this provides another way that L-type channels could be important and use their voltage dependence, very much like EC coupling, which involves the voltage dependence of a CAV 1.1 channel.
Great. Amazing talk. How do you think the local activation of CAV1 in spines with your optogenetic setup can influence phospho-Creb in the nucleus? What can the pathway be?
We think that the engagement of ChemK2 sets off a cascade of events that includes the translocation of a message to the nucleus that is actually carried by calmodulin. Hard to believe at first because calmodulin is so universally important, but there can be a strong disequilibrium caused by the fact that an important beta-driven translocation of a calmodulin carrier can cause disequilibrium and cause calmodulin to frankly go up in the nucleus. That was actually Carl Dijsselroth's PhD thesis. And it was
More
and as I've resigned, stepped down from the chairmanship, my goal is to go through all my papers and find the letter of rejection of that paper or an earlier paper between Dijsselroth and Beto, both of whom you knew from days at Stanford. So far, I've managed to move offices and throw away a lot of things, but I haven't found that letter of rejection yet.
Okay. Lovely talk. I was wondering if there's direct evidence from nimodipine blocking the voltage sensor movement, Ricardo Olcese type experiments, directly measuring voltage sensor movement?
We've done gating current measurements. That was work of Simon Chamberlain with Huda Khaled, and shown that calcium channel blockers do indeed block gating currents. I think John Lutterer was the first person to show that, but that
Okay.
was done in cardiac muscle.
Okay. And then a question for Mike. Very interesting. Local translation in axons, and I assume also dendrites, is sensitive to calcium. Some mRNAs require high calcium for translation, others low. Can you speculate on the relative contributions of L-type channels versus ER or other sources in setting calcium levels in different conditions?
I could speculate, Mike, and we could have a really fun time talking about it, but I don't actually have a knowledge base. I would simply like to say that the link between voltage and calcium and translation is an incredibly important subject. And the tools that you and others have built enable us to ask that question on a transcript by transcript level, on a region by region.
We wanted to see.
It'll be fascinating to find out when translation is important. Is it the end result of transcription, transport of messenger RNAs, or is there an endogenous pool of messages like the ones that Erin studied for CAMK2 itself? What if CAMK2 were the synaptic tag, and the way that it kept acting as a synaptic tag is that the local of translation of CAMK2 kept on providing subunits that could be fed into any protein. So we still have more questions to answer as a field to bring transcription and translation together in a localized way, and to bring biochemistry together, Mary Kennedy type science with electrophysiology, let's say Jeff McGee type science.
Okay, let's see. Two more questions. One from Yingxi. Fantastic talk. How does the amount of calcium influx through the NMDA receptor compare to that through the L-type channels under normal physiological conditions?
About 10 to 1.
10 to 1.
In favor of the NMDA receptor. So if you block each of them and monitor the calcium, and other people like, for example, Bernardo Sabatini have done this, and then you find the NMDA receptor is the gorilla in the room. It's the big source of calcium, and it would make sense for it to be the first landing site for CAMK2.
And
Yeah,
then
that's very interesting. Probably I will hold the question until later.
Oh, let's talk once you get settled in Dallas.
Yeah.
Last question. What if the NMDA receptors do not have the 2B subunit?
Well, I think people think that more NMDA receptors have the 2B subunit than was previously recognized, that there are actually hetero trimers involved. But to my knowledge, that serine 1303, the Strack-Colbrand Kennedy site, is unique to the NR2B site. So,
although in my talk I mainly,
but
All right. Well, I want to thank you and the other speakers. We've eaten up in this webinar. We've spent more time on the questions during the individual session. So I think we should wrap up since we're pretty much on time. So I'd like to thank all three speakers for a fabulous session. It was very enjoyable. And I'd also like to thank my co-moderators, Paul Donlin-Asp and Chao Sun. And I'd like to remind everybody that our next webinar is just next month. It's Dysfunctional Translation in Disease. The speakers are Emily Osterweil, who's at Harvard, Gary Bassell at Emory University, and Giovanna Malucci at Altos Labs in Cambridge, UK. It'll be the same time that you logged in. Check the Lundbeck website if you haven't already registered. We hope to see you there. And again, thanks to everybody. It was really fabulous. And follow up any other questions by sending emails to the speakers. I'm going to send a few emails. Okay. Thank you very much. Thanks a lot.
Thank you. Aaron. Thanks, everyone.
Thanks, Erin. Thanks, Martin.
Be well.
Bye.
Bye.